Good morning, children. I think the last Google Meet, you are not able to get connected to me in a proper way. So I thought I'll summarize the digestion and absorption of lipids once again, and then we'll go into the metabolism. Okay. So I think you know what are lipids now. What are lipids? They are water insoluble fat droplets, and mainly the diet contains different types of lipids, which is mainly constituted by, primarily it is constituted by triacylglycerol, right, TG. 95% of the dietary fat will be made up of TG, and the remaining is constituted by cholesterol esters or free cholesterol, phospholipids, free fatty acids, etc. So what happens to these water insoluble compounds when once they are in the biological system? Once they are in the human system, what is happening to them, or how are they digested? They are digested by a class of enzymes known as lipases, of which again the principal digestion is carried out by the pancreatic lipase, and the mechanism of pancreatic lipase is already understood, I hope, right? Now, how are these lipases bringing about the digestion? They digest the lipids with the help of emulsifiers known as bile salts, right? Bile salts. Bile is synthesized by the liver, but it is stored in the gallbladder, and once the acid chyme or the partially digested lipid enters into the small intestine, the duodenum of the small intestine, what happens? They causes the stimulation of the secretion of both bile and pancreatic juice with the help of different types of hormones like secretin, cholecystokinin, pancreozymin, hepatocrinin, etc. So with the help of all these hormones, what happens? Simultaneously, both the bile and also the pancreatic juice will be secreted into the duodenum, without which the digestion is not possible, because the lipids are water insoluble in nature, and hence when the enzymes try to get access onto these molecules, what happens? They slips away, right? Slip ചെയ്ത് പോകും. അപ്പം അവരെ anchor ചെയ്ത് പിടിച്ച് they have to be made soluble, and this is brought about by the class of compounds known as detergents or emulsifiers, and they are nothing but the bile salts and also the phospholipids. Phospholipids, because they are having phosphate group, no? So phosphates are water soluble in nature, so they are amphipathic molecules. Phospholipids are amphipathic molecules, that is, they are having a polar head and a non-polar tail. So the phospholipids present both in the diet and also in the body, right? Mainly the dietary phospholipids, and also the bile that is secreted by the gallbladder, or what is happening when the food enters? The bile is already synthesized by the liver. If there is any insufficiency, what happens? Hepatocrinin comes into action, and hepatocrinin causes the liver to synthesize more and more bile, right? And this bile will be moving towards the gallbladder, which is an organ present behind or under the liver, right? So it's a storage site of bile. So gallbladder will put our bile is always there, right? Have you seen bile? എപ്പോഴെങ്കിലും നിങ്ങൾ ബൈൽ കണ്ടിട്ടുണ്ടോ? Have you seen how does it look like? It's like a green color fluid, ഒരു ഗ്രീൻ കളറിലുള്ള സബ്സ്റ്റൻസ്. എപ്പോഴാണ് അത് കാണുന്നറിയോ? സിവിയർ വോമിറ്റിംഗ് ഉണ്ടാവുന്ന സമയത്ത് നമ്മൾ ഡയറ്ററി കണ്ടൻസ് ഒക്കെ വോമിറ്റ് ചെയ്ത് പോയി കഴിഞ്ഞാൽ പിന്നെയും പിന്നെയും ഇഫ് ദ വോമിറ്റിംഗ് ഇസ് കണ്ടിന്യൂഡ്, യു ക്യാൻ ഫീൽ യുനോ ഒരു എന്താ പറയുക, കയ്പുള്ള ഒരു ഫ്ലൂയിഡ് പുറത്തേക്ക് നമ്മൾ വൊമിറ്റ് ചെയ്യാൻ തുടങ്ങും. അതാണ് നമ്മുടെ ബൈൽ. ഒരു ഗ്രീൻ കളറിലുള്ള സബ്സ്റ്റൻസ് ആണ്. ഇറ്റ്സ് എ ഗ്രീൻ കളർ. അതിന് ബിറ്റർ ടേസ്റ്റ് ആണ്, ഒരു വല്ലാത്ത സ്വാദാണ് അതിന്. അപ്പൊ അത് നമുക്ക് ഒരുപാട് സിവിയർ വോമിറ്റിംഗ് ഒക്കെ ഉണ്ടെന്നുണ്ടെങ്കിൽ നമ്മൾ അവസാനം വോമിറ്റ് ചെയ്യുക ബൈലായിരിക്കും, റൈറ്റ്? അപ്പൊ ഈ ബൈൽ ഒരു ഗ്രീൻ കളറിലുള്ള സബ്സ്റ്റൻസ് ആണ്. ആ ഗ്രീൻ കളർ എന്താ പറയുക, ഒരു ഡാർക്ക് ഗ്രീൻ കളറിലുള്ള ഒരു സബ്സ്റ്റൻസ്, യെല്ലോയിഷ് ഗ്രീനിഷ് കളറിലുള്ള ഒരു സബ്സ്റ്റൻസ് ആണ്. അതിന് വളരെ ബിറ്റർ ആയിട്ടുള്ള സ്വാദാണ്. അപ്പൊ അത് നമ്മൾ വല്ലാതെ വോമിറ്റ് ചെയ്യുമ്പോൾ നമ്മൾ ബൈലായിരിക്കും അവസാനം വോമിറ്റ് ചെയ്ത് കളയാം. ക്ലിയർ? അപ്പൊ അത് ബൈൽ. അപ്പൊ ബൈൽ എന്താ പറയാ, ഗോൾ ബ്ലാഡറിൽ they are already stored, and whenever there is necessary arises, when the demand arises, what happens? This bile, with the help of cholecystokinin, what happens? It causes a contraction of the muscles of the gallbladder, and it is secreted into the common bile duct, and common bile duct opens into the small intestine. So that is how the bile enters into the small intestine and causes the detergent action. Fats in miscible. The surface tension existing between the water or the aqueous medium of the blood and or the small intestine and the fat droplets will be reduced in the presence of this bile salts, that is sodium taurocholate and sodium glycocolate, and bile in the chain will grip and so that the pancreatic colipase anchor and help him, and with the help of colipase pancreatic lipase carries out the active digestion of lipids, right? So that is how it is happening, right? So small intestinal lumen എന്ന് പറഞ്ഞാൽ ആ വെസൽ ഒരു പൈപ്പ് lumen വെച്ചിട്ട് digestion is happening. They form mixed meshes, right? Those are small disc-shaped molecules. But with the help of mixed meshes, this is small intestinal lumen. This is our enterocyte, intestinal cell in the agatha. With the help of these meshes, what happens is partially digested lipids will be crossing the membrane and enters into the enterocytes. Intestinal brush border agatha clear in the form of free fatty acids 2 mg 1 mg DAG right? With the help of fatty acid binding protein FABP, fatty acid binding protein molecule help they will be entering into the intestinal cells or enterocytes intestinal brush border cross lumen cross it enters into the enterocytes. Enterocytes intestinal juice and intestinal lipase, intestinal juice intestinal lipase they causes a further digestion of 2 mg to 1 mg, and mg will be converted into again free fatty acids and glycerol, right? So that is what is happening. Adhe samayam small molecules anengil adayathu short chain fatty acids allengi medium chain fatty acids okke anengil they are directly absorbed into the a transport phase is happening without the help of any binding protein. Adhe samayam velli molecules okke aanennundengil it requires a carrier protein. They do not require energy. It is by means of active, it is not by active, passive transport, right? That is, it requires the presence of only a carrier. Carrier in the presence of carrier of fatty acid binding protein. Fatty acid binding protein help they enters into the enterocytes. And what happens within the enterocytes that I am coming to explain. Other some short chain fatty acids, medium chain fatty acids are directly enters. They do not require the presence of any carrier proteins. Clear? And from the enterocytes, what happens? These short and medium chain fatty acids can directly enter into the capillaries. Clear? Lumen small meshels untai. These small meshels endinana meshel aayittu irikkunathu because they can be easily transported across the membrane. Now, in membrane, if you permission, bile salt and permission. But once the meshel allows the entry of these lipids into the enterocyte, what happens? The bile salts are removed back. They will be entering, they are released back into the lumen. Ingana release bile acids in the bile salts in the same, they are reabsorbed back by the liver and is taken into the hepatocytes. This we call it as enterohepatic circulation. And the liver gallbladder bile resecrete. So, a small quantity of bile can enter into the large intestine. That's why feces and stool urine excrete type. So, that's 5% majority bile enters back into the portal circulation. Portal vein enterohepatic circulation these bile acids are entering back into the liver and they again stored in the gallbladder. At the food they are entering back into the small intestine. Right? This circulation we call it as enterohepatic circulation. Intestine liver liver gallbladder again intestine are the transport bile acids bile salts simply bile in there. Okay. Right. Other time, or a small amount of short chain and medium chain fatty acids can directly enter into the capillaries because our short and medium chain, very small chains are they can be made miscible. Other time, very molecules it is not possible for them to get smoothly transported into the through the blood. Apart in the chain, very molecule free fatty acids, long chain fatty acids. What happens? They combines back. 2 mg, 1 mg, DAG, that's okay. Free fatty acids join within the enterocytes, what happens? Triacylglycerol will be resynthesized, right? So within the enterocytes, what happens? Triacylglycerol formation is again happening. That is, if the lumen breakdown complete or almost partial breakdown, that's intestinal cells enterocytes they should be transported into the liver for absorption. Absorb absorb if they are remaining like this, they cannot be freely transported. So what is happening? They has to be taken up by the chylomicrons. Chylomicrons in the main constituent in the what are chylomicrons? Chylomicrons are lipoproteins, which are responsible for transporting the lipids. Dietary lipids are vitamin, fat soluble vitamins, cholesterol, phospholipids transport molecules we call it as lipoproteins. There are different types of lipoproteins. The dietary lipids are transported by lipoprotein, mainly known as chylomicrons. Right, so what does a chylomicron do? The major constituent of chylomicron is a TG itself. The major portion, 85% are TG. Now, chylomicron like TG already in diet TG break up. Now, chylomicron form TG should be formed. Now, TG form TAG free fatty acids in 2-MAG, DAG, but diacylglycerol in the chodo. Diacylglycerol in fatty acid would attach it, we get TAG. Clear? About with the help of triacyl, the enzymes, enterocytes, within the enterocytes, there are enzymes which are responsible for carrying out the synthesis of triacyl glycerol, right? So TG will be reformation of TG happens and enters into the chylomicrons. Suppose TG complete breakdown glycerol glycerol TG use up could you follow me? That is, if TGs are present in the lumen after it's completely broken up, what happens? We get free fatty acids and glycerol molecules. So, sadharana glycerol like acyl groups attach ചെയ്യുന്നത് അല്ലേ? അപ്പോൾ enterocytes-നകത്ത് നമ്മുടെ കയ്യിൽ എന്തുണ്ടാകാം? Glycerol molecules ഉണ്ടാവാം. പക്ഷെ ഈ glycerol molecules cannot be utilized for the formation of TG. കാരണം glycerolനെ as such ആയിട്ട് ഒരിക്കലും fatty acidനെ take up ചെയ്യാൻ പറ്റില്ല. എങ്ങനെയാണ് നടക്കുക വെച്ച് കഴിഞ്ഞാൽ glycerol should be first converted into glycerol 3-phosphate. Glycerol 3-phosphate glycerol phosphate attached. So the enzyme is kinase. Now glycerol kinase is an enzyme. Glycerol third position phosphate attached. The problem is that the enterocytes do not have the enzyme glycerol kinase. Clear? Glycerol kinase is an enzyme. Glycerol 3-phosphate cannot be synthesized. Glycerol 3-phosphate synthesize what is happening? 3-phosphate in the phosphate fatty acid attach. So as long as glycerol is remaining as such, glycerol cannot be utilized for the synthesis of TG. Glycerol should be in the form of glycerol 3-phosphate. The problem is that the enzymes enterocytes are devoid of what? Glycerol 3-phosphate. What is the other alternative? Glycerol 3-phosphate. Glycerol 3-phosphate can come from glycolysis. Glycolysis glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can undergo reduction to give glycerol 3-phosphate. Glycerol 3-phosphate is formed for the formation of TG. Digestion glycerol can never be utilized for the reformation or resynthesis of TG because the enterocytes are devoid of glycerol kinase. Clear? So within the enterocytes, what happens? The TG is resynthesized. Resynthesis TG synthase enzyme DAG synthase enzyme the TG will be resynthesized and these TGs are packed into chylomicrons along with cholesterol, phospholipids. Chylomicron apoproteins are ApoB48 and ApoA1. Main apoproteins are ApoB48 and ApoA1. Chylomicrons will be formed. These chylomicrons will be entering into the lacteals, that is the lymphatic vessels, and finally they enters into the thoracic duct. Clear? So this is how the transportation phase happens or absorption of lipids happens. Okay? That is the re-esterification inside the mucosal cells. Inside the enterocytes, the mononuclear cell and free fatty acids are reformed into TG. The fatty acids are activated to fatty acyl-CoA by acyl-CoA synthetase, and activated fatty acids attaches to the mononuclear glycerol to give TAG. This is a picture showing the Michel formation. The already explained TG, lipase fatty acids mononuclear glycerol. These are the phospholipids. So bile Michel formation